People don't realize they have cured cancer in mice so many times. It's not that difficult to do, but when they translate it to the human setting, it doesn't work because of the fundamental differences between humans and mice. We're kind of letting the patients down if we don't speed things up a little bit. Billions have gone into cancer research. You have to question whether for all these billions we've done enough, or have we just generated animal models for just to get better publications? Are we actually helping the patients? And isn't it time we take that bold step and find replacements so we can shorten the time to reach the clinic?
In the last couple of years, I think our models have become more and more accepted. We've published a large number of papers in good journals on those cells and on those models. Then really, we're using a human model, developing that human model, and we're validating that against the human cancer. And if the two match up, then I don't really think there's a need for an animal. Really, we need the drive for people to spend more time, more effort on developing the models so that scientists can then use them to investigate human disease.
You can take a cell and you can look at every single gene and you can find every single mutation that it has, which is very, very powerful. So if you can apply that to a patient who comes in and they have a tumor of one sort, you could potentially take just a skin sample and you could tell them, yes, you have this many mutations. The potential is there, and you can see in a few years' time when these type of technologies become a lot cheaper, and then you have personalized medicine. So you can't really do that with a mouse.
As part of the Animal Replacement Centre, we're making sure we build a really robust range of different animal replacement models. We're not just relying on one model. Using human models in vitro because it's very reproducible, very controllable model, and it's also very clinically relevant. It's relevant to humans. This is a human model using human cells. And so because it's got that direct relevance, you can start exploring important clinical questions directly, like you can't necessarily do in animal models, which also have the problem of being much harder to control, harder to reproduce. So this is really where I came into this direction. I wanted to do something reproducible, which is clinically relevant, and where you could control the experiments so that you could actually get meaningful outcomes.
The key thing that will cause a change is if there is alternatives that can be even superior to the use of animal models. You cannot resist that advantage. It's cheaper, it's more closer to the human condition. The time to reaching the clinic will be vastly reduced. It's not about, we just hate the use of animal models and we're trying to stifle research. The opposite. We want to accelerate scientific research. And you do that by developing models that are very close to the human setting, not add this extra intermediate steps with the use of animal models.
So I would like to thank all supporters who carry on the excellent work in supporting Animal Free Research UK, and without your support we could not do the research that we're doing in our laboratories. So we are truly appreciative of your support and help in enabling us to do the research that we are carrying out.